for joining us. We'll get started in just a minute. We'll give folks another minute or two to get connected. Okay, let's go ahead and get started. Thanks for joining us this evening for the Nebraska Equine webinar series. I'm Jessica Peterson. I'm an assistant professor in the Animal Science Department here at the University of Nebraska-Lincoln. And specifically, I'm in the Animal Breeding and Genetics group, and part of my research focus is to study equine genetics. So the presentation that I'll be giving today is about genetics and genetic disease of the horse. If it works. So the topics that I plan to cover, first we're going to start with an overview of genetics and inheritance, just to get everybody on the same page of how genes are transferred and what information we can gain from genetics. I'm going to spend a little bit of time on how genetic tests are developed. I think this is important as users of these genetic tests that you understand the background and the scrutiny that these tests go through before they're offered to the public. And then finally, we're going to talk about current genetic testing for disease traits in horses. So some of these you might be familiar with. Some of these are newer genetic diseases. And we're going to go through a pretty good list of these. The goal is not for you to be experts on any of these diseases, but to just have an idea of what is out there. So if you are perhaps breeding a particular type of horse, you're aware of what potential genetic tests you should be considering in your breeding program. So if you have questions, go ahead and use the chat to ask them at any time. I won't address them as they come up. I'll wait till the end and address questions, but I'm happy to do that. So make sure you take notes of questions that you have as we go through this. Okay, so this kind of complicated but interesting picture that I have up is what we call a phylogram, and this is showing how different breeds of horses are related to one another. And so horses that are more closely together, such as the Shetland and the miniature up here on the top left-hand side of the screen, they're more closely related than, say, the Shetland is to the paint or the quarter horse, because they're more distant on this tree. So this data, this tree was created using genetics. They're using DNA markers showing how similar these animals are to one another. And not surprisingly, if you look at the patterns here, you see that the horses in this area are Scandinavian breeds, so they share a similar geography. That way they can interbreed with each other more easily. Down here we have our draft breeds, so they're not surprisingly more closely related to one another than they are to the Scandinavian breeds. And then we have our US developed breeds, we have our paints, our quarter horses, and our thoroughbreds over here. So what's interesting about genetics is not only can we determine how animals or breeds are related to one another, we can then use this to find differences. What makes these animals different from one another? Whether it be a quarter horse from an Arabian, or whether it be a horse with a particular condition or disease from a horse that doesn't have that condition or disease. So let's do a very little bit of basic genetics. Remember that our DNA is inherited from each parent, and that in every mating, 50% of the parent's DNA is contributed to the offspring. So if we say that this horse up here on our left is our sire is going to have 2 copies of every chromosome. He inherited one from his dad and one from his mom. Likewise, the mare has 2 copies of all of her chromosomes. She inherited one from her mom and one from her dad. So if these two horses are mated, that stallion is going to donate one of his chromosomes of each type of chromosome to his foal. The mare is going to donate one of each of her pairs to the foal, and therefore that foal is a representation of genetics of both the mare and the stallion. So I'm going to introduce some terms that I'll be using throughout the talk tonight. First is allele. So a variation of DNA at any point of interest, that could be a gene, it could be any region of DNA, we call that an allele. So an allele is a variant. And at each of these points of interest in the genome, every individual has one allele from the sire and one allele from the dam. And the combination of those two alleles is what we call a genotype. So an individual's genotype consists of what they inherited from their sire and what they inherited from their dam. So in this example, we consider our stallion, and let's say I'm interested in this particular locus of the genome. This might be a gene that I'm interested in. So I'm going to say that that stallion has two copies of the same allele, and I'm going to use a capital A, or I'll say big A, to denote what variety or what kind of allele that is. So that stallion has alleles big A and big A, and therefore his genotype we say is big A, big A. The mare, on the other hand, she has two different varieties of that allele. She has a little a. So she has two little a's, and her genotype therefore is little a, little a. However, when you would mate these two horses and you have a foal, and because they have two copies of the same allele, we call those horses homozygous. So homo meaning the same, and our zygous is referring to that allele state. So the stallion only has one type of allele, but he has two copies of it. They're the same. Same with the mare. And now we get to their foal. That foal inherits the big A allele from his dad, inherits a little a allele from mom. Therefore, that foal's genotype is big A, little a, and we say that foal is heterozygous. So hetero meaning different, and our zygous, again, referring to that allele state. Okay, so again, mom and dad contribute half of what they have to that offspring. So some other terms that we need to review is dominant versus recessive. So simple traits, and most of what we are going to talk about today are simple traits, can be described as dominant or recessive. So if you just joined, can you please mute your microphone? We're getting some feedback. Please, thank you. Okay, so simple traits can be described as dominant or recessive. A dominant trait does not have to be the one that's most common, and I see that misconception a lot. But a dominant trait is also not necessarily the healthiest, but it could be. It could be either or both of those. So for example, the BRCA1 allele, so the allele that is implicated in many types of breast cancer in humans, that is actually a dominant allele. So that obviously is not the healthiest, and it's also not the most common. Fortunately, a majority of people do not have this allele. Another example I like to use in class is polydactyly in humans. So most people are familiar with cats that have multiple or have extra toes. This can happen in humans. And in one case it happens in humans, it is also a dominant allele. And so far I've not had a class where a majority of the students have more than 5 fingers on either of their hands. So a dominant trait does not have to be most common or healthiest. It could be. But what dominance does describe is how that allele is expressed in our individual. So we want to think about that dominance as a definition of how that allele is expressed. So I'll stick with an example in humans. So remember that every person or every horse has 2 alleles for every gene, one from mom, one from dad. And each of those alleles carries information about whatever trait we're interested in. So in humans, eye color, if we simplify it, is determined by 2 possible alleles. Brown, which I'll use a big B to denote, and blue, which is a little b. The brown allele happens to be dominant, and the blue allele is recessive. So in our definition of dominance, if a dominant allele is present, that allele determines the phenotype or the characteristic that is seen. So if the genotype is big B, little b, that big B allele, because it's dominant, it covers up that little b allele, and therefore the person has brown eyes. The recessive phenotype, or blue eyes, is only observed if that dominant allele is not present. So the only way you have blue eyes is if you have two little b's, little b alleles at that particular locus in the genome. So if you had a big B, you wouldn't have blue eyes, you would have brown eyes. So this is important because we can use this in horses a lot. If we understand the relationship between our alleles, whether it's dominant or recessive, we can often infer information about the parents. So for instance, if both of your biological parents had brown eyes, but you yourself have blue eyes, you can learn something about their genotype. If they have brown eyes, they must have a big B allele. But if you have blue eyes, you must have inherited a little B allele from each of your parents. Therefore, your parents' genotype must be heterozygous for big B, little b. So we can use outcomes of matings in our horses to identify what alleles individuals might be carrying that we can't otherwise see. So to finish out our examples of basic genetics, I want to use coat color. Because coat color is fun to think about. It's fairly simple, and it illustrates a lot of the things that come into play when we talk about disease as well. So often I'll ask students, I'll say, how many colors are there of horses? And just on the screen right here, I see 8 different colors. So I would register these horses as 8 different colors. However, genetically, horses come in 2 base colors. And those 2 colors are determined by simple dominance. All other colors that we see are simply modifications of those 2. So here are the 2 colors of the horse. Red, which we'll call sorrel or chestnut, depending upon what breed you grew up in, and black. So those are the 2 genetic base colors of the horse. And this is determined by a gene called MC1R, or melanocortin-1 receptor. So when we go back to our example, as brown eyes was dominant to blue eyes, black, the horse coat color, is dominant to red. Or we could think about it and say that red is recessive to black. So the alleles at this locus are big E, which denotes the dominant allele for a black horse, or the little e, which is a recessive allele that red horses will have. So we have this horse here who is obviously black in coat color. Because black is dominant, the only way to have a black horse, or for that horse to be black, is to have a big E allele at the gene MC1R. And so therefore, since we know he has a big E allele, we know his genotype has to be either big E, or big E, little e. If we consider our red horse, because black is dominant, the only way that that horse can be red is for that horse not to have a big E allele at the gene MC1R. So in this instance, this horse has to have the MC1R genotype little e, little e. If she had a big E, she wouldn't be red. So finally, we already said that this horse has to have a big E allele at the locus MC1R to be black. But without more information, we don't know his complete genotype. So if that's the case, I often write this with a big E and then an underscore, saying that we know he has to have a second allele because he had 2 parents and he's a healthy horse. But we're not sure what that is. It could be another big E or it could be a little e. Regardless, that horse is still going to be black. So that's an example of how recessive alleles can hide from one generation to the next, which can be a good thing or it can be a bad thing. Okay, so now when we go back to this, we know now that horses come in 2 base colors. They come in red or they come in black. And it happens that the 4 horses on the left side of the screen are red genetically, whereas the 4 horses on the right side, I think I said that right, 4 horses on the left are red and the 4 horses on the right are black. And the way we can tell that most quickly is that black horses, or horses with one of those big E alleles at that locus MC1R, have a black mane and tail and black points. So black legs if they're not covered by white, and black tips of their ears. All other colors are modifications of those 2. And we won't go through all these, but here are some of the modifications. We have a gene that turns black into bay. We have dun, cream, roan, all those white patterns. But what I want you to think about is that all horses have the same set of genes. They just don't necessarily have the same alleles at each gene. Excuse me. So even a chestnut horse has a gene that causes dun, but it just has a recessive genotype, so that dun color is not apparent in that horse. So the horse on the left, it has a genotype at MC1R of little e. That means it's a red horse. And it has a copy of the dominant dun dilution, which makes that horse a red dun rather than a sorrel. And the dun, you can see it has tiger stripes on his legs, and it should have a dorsal stripe down his back. However, the horse on the right, it's also a sorrel horse or chestnut, has little e genotype at MC1R. It still has the gene that would code for dun. It just has the recessive genotype, so 2 little d alleles. That mean that dun color is not observed, and that horse therefore is just chestnut. So horse color, like I said, is usually a fun example to start thinking about genetics. So horse's color or any trait depends upon a combination of all those genetics considered together. And horse color is one example of how it can get complicated pretty quickly. So this particular horse has a black base coat, it has a big E allele, it has an agouti dominant allele, it has a cream dilution, it has a dun dilution, it has Tobiano, and it makes that horse a dun-skinned Tobiano. So what you see, whether it's coat color or whether it contributes to disease and performance, is a combination of all those genes and all the horse genetics put together. Okay, so let's move on to what you're all here to hear about, which is genetic disease. So genetic tests are becoming more and more common and fairly easily accessible. So let's talk about how those genetic tests are developed. So usually the goal is to identify an allele or alleles that cause the disease in the horse. We assume that those alleles, particular alleles, are present in the horses with the disease, but they're absent in healthy horses. So the challenge is variants in DNA are not hard to find. So I recently sequenced the entire genome of 6 horses of the same breed, and I found over 14 million variants among those horses. So if I was looking for variation, I found it. There's 14 million of them that I could study. Some of them are shared between horses, and some of them are unique to individuals. So this is what I'm looking for. I'm looking at DNA sequences, and I'm looking for regions that it's different. So those are easy to find. So while variants are easy to find in common, associating those variants with the phenotype or the trait that you're interested in is very complex and definitely not straightforward. One of the reasons is because conditions that appear similar might be due to different causes. And an example to think about is lameness. You can probably think of half a dozen different reasons a horse might be lame. He could have an abscess in his hoof. He could have broken his leg. He could have a stifle injury. He could have a suspensory injury. So if we're just studying lameness, we're going to doom ourselves to failure by considering all those things together. So the first step is for whatever disease or trait you're trying to study, you need to have an accurate means to diagnose that trait. And veterinarians are extremely important when it comes to identifying genetics associated with disease. Often we look for very specific ways to make sure that the horses we are studying with the disease do indeed have the exact same disease. And similarly, we want to find horses that we are very sure do not have that disease. So our diagnosis is very important. So our second step is actually not even as hard as the first step anymore. We have a lot of genomic tools where we can identify alleles and variants or genotypes that we can associate with the diseased horses that aren't present in the non-diseased horses. Once we find that subset of variants that seem to be present in diseased horses but not in healthy horses, then we have to look at which ones we have a reason to think might alter the biology of that horse to result in the condition that we're seeing. So is that variant in a gene that we know changes the immune function of the horse? If so, it might be the one that's contributing to our disease. And finally, we need to validate the association between those genetic variants or the variants and the phenotype. So what do I mean by that? So reliable genetic tests are tests that are validated. Those tests have been described or demonstrated to alter the biologic function in a way that causes the condition that you're studying. So this can be through other functional assays. This can be through in silico predictions, but you need to describe how these variants might alter the biology of the horse. Then you need to further demonstrate that that variant causes that condition in horses other than the ones that you originally studied. So for instance, maybe my original study was a family of 25 quarter horses, and I found a variant that seems to be associated with a particular disease. Then I need to go and look at other horses outside of that family that have that disease and see if they have that variant as well. If my test is reliable, that will be the case. I also need to go out and see how frequent that variant is in the general population, either that breed or crossbreeds. And that will give us an idea of one, whether or not it's real. If the disease is very harmful, if it's fatal, or if it has severe consequences, and I find that variant that I think causes it is at high frequency, I probably don't have the right variant. And further, having an idea of the frequency across the breed gives us an idea of how worried we should be about it when we are breeding horses. And finally, a validated test is published in the scientific literature, and that happens after it is thoroughly peer-reviewed by other researchers and scientists. So this gives other researchers information they need to go out and test horses and test the hypotheses in that paper to see if that test is truly correct, and to further study the biology so we can learn better ways to maybe treat horses with a particular disease or to prevent it. So the tests are the genetic diseases, all of which have a test associated with them that you can purchase, have gone through this process. And I will give you the citation for the publication and also information on the biologic consequence of each of these mutations. So I'm going to start with the AQHA 5-panel test. You might be familiar with this. I think it's worth going over most of these because they are fairly consequential and prevalent. These are all diseases with simple mode of inheritance, so they're either dominant or recessive. Tests are required now for breeding stallions in the Quarter Horse Association and recommended for brood mares. Since you have this information, or you can access this information, it's important to not be passing on traits that you don't want in the next generation. So HYPP is probably the most well-known genetic disease in the quarter horses. HYPP stands for hyperkalemic periodic paralysis. This is a dominant disorder of the gene SCN4A. Because it's dominant, if your horse is a heterozygote, has one copy of the disease allele, which they denote as an H, or two copies, it is going to have symptoms of this disease. So the consequence of that mutation in that gene impacts the balance of sodium, potassium in the muscle cells. So the muscle cells are hyperexcitable, and their contractile properties are not normal. HYPP is interesting because it's one of the few genetic diseases that can be attributed to a particular bloodline. So all horses with this HYPP mutation are descendants of Impressive, who was a 1969 quarter horse stallion. He's very popular in the halter bloodlines, and he had good muscling. However, I do want to mention that there's no scientific evidence that HYPP itself increases muscle mass in these horses. So I would argue that there isn't a reason to keep breeding HYPP horses. So symptoms, horses, if they have this disease, they can show uncontrolled twitching of muscles. It can be mild, it can be a nuisance, or it could be severe. These symptoms can be decreased by diet. And people will ask, what do people mean when they say they have a non-symptomatic horse? You can have non-symptomatic horses in that the horse might not have an episode, or you might not observe it have an episode. Unfortunately, though, you can't predict when these may or may not take place. And I've heard enough stories and talked to enough people that unfortunately the first episode they knew their horse was having was fatal. So unfortunately, that's what we're dealing with HYPP. So the outlook is that heterozygotes often can have a useful life. Many, maybe not many, but they may suffer an early death due to this disease. Whereas homozygote horses, the HH, often have more severe symptoms. So what is the prevalence of HYPP? And actually it's not, if you consider the quarter horse and paint horse populations as a whole, it's not extremely prevalent. About 1% in quarter horses and less than 3% in paints. However, if you break up quarter horses into these performance groups that are popular, you'll see that almost a third of halter horses do have the HYPP mutation. So that's important to think about if you're considering breeding halter horses. So the second 5-panel test that I'm going to talk about is PSSM. It stands for polysaccharide storage myopathy. Again, it is a dominant disorder, and the gene that's implicated in this case is GYS1. It's glycogen synthase. So if you have a horse that's heterozygous for the mutation, they use a big P to denote the mutation, or homozygous for it, your horse is affected by this disease. What happens if your horse has this mutation is that this gene causes an excess of abnormal sugar to be stored in the muscle. So this form of the sugar, this glycogen, is not as easily used for exercise. Horses need glycogen as a source of energy for muscle contraction and muscle metabolism. When a horse has this mutation, they can't mobilize that energy source properly. And so the consequence is that after moderate exercise, those muscles can't function properly and they will cramp up. So the symptoms are tying up with exercise, or those muscle cramps. The horses will also sweat, they may stumble, and it is painful to the horses. This is usually first noticed at about 2 or 3 years of age. Not coincidentally, this is when horses are generally start to be worked. And the outlook is that performances of affected horses can be managed by diet and exercise. You want these horses to be exercised frequently. You also want to have them on a low carbohydrate diet. And while PSSM isn't a death sentence by any means, it could limit usefulness of high-performing individuals. So this is a really interesting disease in that it's not just a quarter horse or paint horse disease. And actually, if you look at the frequency, it is very, very frequent in our Belgian and Percheron draft horses. Over half of the Percherons that were studied were found to have this particular mutation. And even in quarter horses or paints, it is pretty prevalent. And my take-home message about PSSM is you probably do know an affected horse. And this is my mare, and unfortunately, she is one of the horses that has a copy of this mutation. Within the quarter horse breed and paint horses, it's actually more frequent than HYPP. I think it's lesser known, but it happens to be more frequent. In this instance, it also is found more commonly in halter horses. And if you think about it, they're not subjected to the same performance pressure as the other groups. So this mutation probably doesn't impact their performance as much. But it's also found across the working cow horse types and a little bit in the racing and barrel racing horses. So Dr. Stephanie Valberg is at Michigan State University, and I would argue that she is if not the expert on equine neuromuscular disorders. She gave a webinar actually in November of last year that's recorded on My Horse University. So if you want to learn more about PSSM, I'd highly, highly recommend that you listen to her webinar. So continuing with the 5-panel test, HERDA is next on the list. HERDA stands for Hereditary Equine Regional Dermal Asthenia. And as that name suggests, the dermal part of it is a disease of the skin. The skin is loose and fragile, and it's due to a recessive mutation, the PPIP gene. What this gene does is not completely understood, but it alters how collagen is folded and the strength of that dermal tissue. So collagen is very important in connective tissue and in the epidermis, and this gene is altering its function. So usually your skin, if you pull skin on your horse, it should snap back, it should be elastic. In this case, it isn't. The skin damages very easily, and hematomas are common. People usually first observe this about 1.5 to 2 years of age. Like PSSM, again, this is when horses start being worked. Many times it's when somebody goes to cinch up a saddle for the first time and they're putting stress on that skin. This is not a good thing, as you can imagine. Unfortunately, the affected horses are usually euthanized, The reason is that these skin lesions open the horse up to infections, and we all know that horses have a skill at injuring themselves. So if they're scratching on a fence post, or if they get in a little fight with their pasture mate, they could have these hematomas or damage in their skin. But because it's recessive, we can avoid it by not breeding two carriers together. So if a horse only has one copy of this mutation, that horse is perfectly fine. You just don't want to breed it to another horse that carries a mutation to chance an offspring being affected. So HERDA is prevalent in quarter horses and paints at a fairly low frequency. And in this instance, it's not in the halter horses. This is commonly found in cutting horse bloodlines. So about 14% of cutting horses were found to have a copy of this HERDA mutation. So the final disease in the 5-panel test that I'm going to go over today is GBED. It stands for glycogen branching enzyme deficiency. And it's a defect in glycogen metabolism. So as in PSSM, glycogen is a very important energy source for muscles. So if a horse has a recessive genotype for this gene, the enzyme that is needed to create this glycogen in a form that the horse can use is defective. So it can't store or use glycogen properly. So if the horse can't get energy to its muscles and to its other tissues, that's obviously a problem. Unfortunately, foals with the recessive genotype are often aborted or stillborn, and foals that do make it to be born alive will show weakness, seizures, and cardiac respiratory failure, and unfortunately, this is fatal. So again, it's recessive though, so it's important to test your horses, especially if they're of a certain bloodlines, in quarter horses or paint horses, so you don't breed two carriers together. So where is it in the quarter horse population? It's fairly frequent, 5%. If we look at performance groups, it's most common in the Western pleasure types, but it is found in most of these other performance types as well. So that was the last of the 5-panel tests that I'm going to talk about. I'm going to go through some other known genetic disorders. They're across different breeds, and then I'll end up with some of the newer ones that have just come out. So if you breed Arabians or interested in Arabians, SCID is probably familiar to you. Severe combined immunodeficiency. As its name suggests, it's a dysfunction of the immune system. It is found in Arabians and it is recessive. As a consequence of this mutation, horses with it cannot produce immune cells that they need to fight off infection. And so after the foal is about 5 months old, when its immunity is from its mother is past, that horse itself cannot fight off infections if it becomes ill. So often this leads to illness, which eventually will lead to death because these horses just cannot deal with pathogen challenges. And it's estimated in the Arabian population that 8.4% are carriers of this. So again, it's recessive. So it is fatal, and it's important to test Arabian breeding stock to make sure you're not breeding two carriers together. So continuing with the Arabian trend, cerebellar abiotrophy, or CA, is next. It is a neurological disease. It also is recessive, and in this case, it's a mutation in the gene called MYTYH. And this gene is usually active in repairing damage to neurons, and specifically cells that are called Purkinje cells in this cerebellum of the brain. If this gene is mutated, if you have the recessive genotype, the brain can't repair itself properly. And that portion of the brain is related to coordination. So symptoms are tremors of the head and neck, lack of coordination, and abnormal gaits. So I was going to link to it, but there's some pretty good videos on YouTube, of course. One of it is this foal in this picture, and he has almost a head nodding characteristic. He's a little bit uncoordinated and can't quite control and steady his head. The problem is, as these horses get older, they still don't have good balance, and they can be a danger to themselves or people handling them as they stumble. The frequency is fairly high in Arabian horses, so again, it's recommended that these horses are euthanized. They don't pose a significant risk for injury to themselves or to others, but we can avoid it if we don't breed two carriers together. Sticking with the Arabians, lavender foal syndrome was described in 2010. Again, this is a neurological disease. This is primarily in Egyptian Arabians. It again is a recessive mutation of a myosin gene. The symptoms of this are like its name says. The foal is born alive, but it's born a dilute color. And if you're there at foaling, you might think, wow, this is really unique, and I have a really fancy looking foal, because Arabians aren't supposed to come in dilute colors, right? But the problem is that this color, dilute color, is associated with these extreme neurological deficits. So that foal immediately will show seizures, can't control muscles in his limbs, and they won't be able to stand or thrive. So unfortunately, this is fatal, and approximately 10% of Egyptian Arabians are estimated to carry this particular mutation. So again, we can avoid it by not breeding carriers together. So I'm going to switch away from Arabians. I'm going to talk about JEB, junctional epidermolysis bullosa. And this is interesting because this disorder is in several different breeds. It's in Belgians, other drafts, and also saddlebred horses. This is caused by a recessive mutation, whether it be in Belgians or saddlebreds, but it's of two different genes, depending upon which breed that we're interested in. These two genes, however, are in the same gene family, and they have very similar functions. So what these genes do is they help build a basement membrane underneath the skin that attaches the skin basically to that horse's body in a tight manner. So symptoms are these very disturbing skin lesions and blisters. Skin will slough off at pressure points. Hooves can be lost, and their mouth also has some abnormalities in their teeth and how their dentation is developed. So this foal in this picture, this poor little foal, this is the pictures that you see of this. It commonly shows that the front legs have absent of any skin, and I would suggest that it's probably due to an attended foaling where somebody is trying to help this foal be born, and putting pressure on those front legs as it came out and didn't know that it had this condition, and in effect pulled skin off of that foal. People have tried to treat these and tried to keep these horses alive and thriving, but as you can imagine, a lot of infections can occur with these big open wounds. So it's generally fatal. These horses are euthanized pretty early. So the frequency of these mutations are about 70% in Belgians and 5% of saddlebreds are carriers. So again, don't breed the carriers because we definitely don't want foals to have this issue. This is a fairly new genetic test that you can order. This is hoof wall separation disease, and this was found in Connemara ponies. So it's a disorder of hoof structure because hooves easily break and they crack. So anytime that there's force put on that hoof wall integrity is low, and it will break. So it's due to a recessive mutation of SERPIN-B11 gene. And the symptoms, as its name suggests, as I said, there's severe cracks or breaks in the hooves. They usually start showing up in the first 6 months of life, and with those severe issues in the hooves, that causes lameness. There's some management that can be done to help reduce the severity of this, but it's generally obviously not a good thing. The frequency is about 14% of Connemara's are carriers of the mutation. And so the outlook is don't breed two carrier horses together again. This is hot off the press, and this is a little bit different than what we've been talking about previously. So ocular squamous cell carcinoma is a cancer of the eye, and it's not a dominant or recessively inherited disorder. However, risk factors were found in a gene called DDB2. And DDB2, which is hard to say fast, is similar to the BRCA1 gene I mentioned earlier, in that it acts in DNA repair. And so in particular, what DDB2 does is it repairs damage due to ultraviolet light. And so this mutation impacts the ability of this gene, and the protein of this gene, to identify damaged cells to go and fix them. If you can't fix this DNA damage, it leads to cancer. So symptoms are mass in the eye, as you can see in that picture. Those masses usually grow pretty quickly, and they can lead to blindness. So sometimes they can be removed and you can keep an eye on it, pun intended, or other times they can unfortunately invade other cells in the eye and lead to blindness. So frequency is pretty high in halflingers and in Belgians. And Belgians and Percherons are pretty closely related to halflingers genetically, so that makes some sense. So what they suggest you do is that while you don't necessarily want to not breed horses that have this risk factor, if you know they have it, you can make sure that you are monitoring those horses closely. You can have your veterinarian give them an eye exam more commonly than you would otherwise. And it's suggested that you provide protection from the sun. So a fly mask that has some UV protection will help DNA damage from occurring from the UV light. So even if they have this mutation, you hopefully hedge your bets that you're not going to have to deal with it. Okay. So coat color, which we started talking about, has some interesting connections to disease. And I want to go through a few of those. So ovarolethal white foal syndrome is one you may have heard of. It's in paint horses and in paint crosses. And it has something to do with this white color pattern called overo. So this horse on the right is an overo. He's called a frame overo actually because it looks like he has a picture frame around him. So dark color goes across his back, down his legs, and kind of in a rectangular pattern. So overo itself is dominant. So you only need one allele of the overo variant to cause this cool white coat color pattern. However, lethal white is recessive. So if you have two alleles, you will be overo, well, you will not be overo, you'll actually have a lethal condition called lethal white. So one is good, especially if you like that fancy white pattern, and two copies are bad. So the gene is EDNRB. And like I said, it's most commonly found in those frame overos, like this horse in this picture. However, it can be deceiving because some horses can have this overo mutation and not show a lot of white. So it's important that horses are tested for this if they have paint lineages, and even some of the quarter horses, because they could be carrying this and you're not aware of it. Also, some of the Tobiano horses may also be carrying an overo mutation. It's hard to see sometimes with all the different white patterns. So it's recommended that you test your stock horses, your breeding stock, if you're considering breeding them, especially if you're going to breed them to a horse that you know is overo. So here's a picture of a mare and a lethal white foal. The foals are born usually all white. Sometimes they do have a little bit of color on them. So the foal is born and seems perfectly normal, although all white, but after a little while it will show signs of colic. And that's because the intestine is not formed correctly. It's very similar to Hirschsprung's disease in humans. So unfortunately there's nothing you can do about this, and it will be fatal. So it's recommended that you euthanize these foals as soon as you're certain that is the condition that the foal is suffering. So again, because it's recessive, you shouldn't have to deal with foals with this. You can use genetic testing and test your horses. And again, you should probably test all of your paints and quarter horses, even if they are not visibly overo, because it can be carried in horses that don't show a lot of white color. Appaloosas are really interesting when it comes to genetics. So Appaloosa color has been associated with different eye conditions. So let's talk about Appaloosa coat color patterns, or leopard spotting. It's determined by an incompletely dominant gene. That gene's called TRPM1. It's a little bit different than the dominants we've been talking about to this point. So if you're homozygote for the recessive genotype, so little LP, the horse doesn't have any spots. So in that top row of the picture, those are solid color horses, they don't have any spots. However, if you're a heterozygote, have one copy of the big LP and one copy of the little LP, the horse has spots. So it's pigmented spots. And all three of those horses in the middle row, you can see the dark spots across their body. The distribution of where those spots are depends upon another gene, but we're just interested in the spots right now. And if a horse is homozygous for the big LP genotype, it's almost like those spots go to excess, so the spots don't even have color themselves anymore. So there will be white with no spots. So clear spots, if you kind of want to think about it that way. So there's a 3 different levels, I guess, if you will, of leopard spotting in Appaloosas. So what does that have to do with disease? So this particular spotting gene, TRPM1, is also linked to congenital stationary night blindness. So similar to the lethal white, whereas having 1 copy of the big LP is going to give your Appaloosa horse spots, if you have 2 copies of it, you're going to have the white spots, but you also will have to deal with this congenital stationary night blindness. These horses have impaired vision in the dark. However, it isn't progressive. So it's not going to get worse as they get older. Often the horse can deal with it pretty well. You just want to be aware. If your horse does have this condition, if you're going out in a trail ride and it's getting towards dusk, just be a little extra careful and don't point your horse over some terrain it's not used to. However, this gene also has been linked to equine recurrent uveitis. So this is interesting in that it, again, is a risk factor. Like the squamous cell carcinoma, the eye cancer, if your horse has a copy of the big LP or 2 copies of the big LP alleles, it contributes to risk for uveitis. This is also called moon blindness. And in this case, inflammation occurs in the eye. It can often be treated, but it does reoccur, hence the name recurrent uveitis. And if it's not treated regularly, it could lead to blindness. So if you have an Appaloosa horse that has the big LP allele, it doesn't mean your horse is going to get this. It just means your horse has a higher risk of this than a horse that doesn't have that allele. So it's something that if you have a Appaloosa horse with spots, you know that the horse has at least one copy, that big LP allele. And so I would just be a little more conscientious about checking that horse's eye and making sure if it shows any signs of inflammation that you get a vet out and have it treated. So that's a good lead into simple compared to complex traits. And we're going to wrap up here pretty quick. Most of what we've been talking about are simple traits. So they're dominant or they're recessive, and they're largely determined by one particular gene or one particular locus of the genome. But most of the things that we're really, really interested in are complex. So they involve many, many genes, as well as the influence of the environment. And by environment, I don't just mean where the horse lives, but I mean how that horse is trained, what that horse is fed, and all those other things that go into making a horse a horse. So one of those things I mentioned that is complex is equine recurrent uveitis. We know that TRPM1 is a risk factor. However, it also appears that another section of the horse's genome contributes risk to whether or not the horse is prone to developing this problem. Metabolic syndrome is something a lot of people are very interested in studying, but also is very complex, although it likely has at least partially a genetic background of what causes it. Different performance traits, nutritional needs, and finally susceptibility to disease. This is becoming more and more important. If we can breed horses that are less susceptible to disease, I think we'd all agree that that's a good thing. So understanding these complex traits using genetics and further using genetic tools for breeding decisions is possible, and I would say it is in our future. We are going to get there, where we're going to use information on a horse's full genome to determine best methods of feeding that horse, best ways to manage that horse for that horse's optimal health and performance. So finally, when we think about genetics and what role it plays in the horse industry, testing is now available and allows for informed breeding decisions and provides a tool for veterinarians as well for diagnosis of horses that are presenting with symptoms similar to some of these diseases. I believe that genetics should be considered along with the horse's disposition, with how it's trained, its conformation and bloodlines. It should be one of those factors that you weigh in when you're looking to buy a horse. However, the genetic tests that you use in your decision making should be validated and provided through a reputable testing laboratory. So I think it's important to know that genetic test that you're using to base a decision on, whether it's a health decision or a purchase decision, is something that you can feel confident is telling you what it says it is. And also that many conditions aren't genetic. So we can't blame genetics for everything, and maybe not even for most things. It's very important to have a good veterinarian and evaluate your horse's condition before you start going off and trying all these genetic tests. Veterinarians are highly trained in diagnoses, and they're important in making decisions for your horse's health. So that said, my take-home message is be informed about genetics, but enjoy your horse. We spend a lot of time worrying about little things for our horse, and sometimes forget that we should just be enjoying that horse's company and that pleasure our horse provides. So with that, I think we have a few minutes here, and I'll be happy to take some questions if you have them. I think if you have a question, there should be a, on the more button above, there should be a chat. You can type your question. If you don't have any questions, that's fine too. You can find me on the website at unl.edu. Again, my name is Jessica Peterson. I'm in the Animal Science Department. And I'm always happy to talk about horses. I'm one of those crazy horse people that enjoys talking about horses and also genetics. So anything that comes up, I'll be happy to share what I know about it with you. All right, everybody's being very shy. So everyone have a good night, and thanks for attending.